All right, this is part 2 of the immune system on the adaptive specific host defenses. All right, so a quick overview. This is what we started with in the last lecture. Remember, I mentioned that most people are familiar with the side we're about to talk about. That's on the right-hand side in the little picture. It was the left-hand side that most people don't really think of as their immune system. So again, we have a multi-level network of innate, non-specific protections as well as adaptive specific protections that we refer to as the first, second, and third lines of defense. This chapter is going to be on the specific adaptive defenses. So there's some old names for the specific adaptive side to all of this that we still use. We talk about there being the humoral side and the cellular side, so we'll be delving into that a little bit more. So an overview of what we'll be doing, and of course our objectives. So let's look at some basics about the adaptive immune system. It is specific, so it has specificity, which is not an easy word to say, meaning it targets specific pathogens. So we were looking at innate in the last lecture. It doesn't really, it doesn't have any way of recognizing. It sees a microbe and it doesn't know whether it's good or bad. It's supposed to be there, not supposed to be there. It doesn't matter. It will get rid of it. This side actually knows. This side can target specific pathogens. It also has memory, which we didn't see before. Memory allows it to make like a little chip of whatever that pathogen was in the past and how it fought it, so that it can pull that back up quickly if it encounters that pathogen again, which explains why with a lot of illnesses, they always say you only have to get it one time and then you're immune to it forever. And that's exactly why you have these little memory chips of how your immune system fought that particular pathogen, and you come in contact with that pathogen again, and what it immediately does is pull that information up and can mount an immune response much quicker. Primary response is the first exposure to the pathogen or the vaccine. When you're exposed to the pathogen, typically you get sick. When you're exposed to the vaccine, you don't necessarily get sick, and we're going to talk about vaccines at the end of this lecture. The secondary response is any subsequent exposure to a pathogen. So there's not a tertiary or a quaternary or anything like that. Every time after the primary response that you are exposed to that pathogen again, we refer to that as the secondary response. That's when that memory chip kicks in, and most of the time you don't even get sick. You don't even know you were exposed. So let's just use the example of the chickenpox. I know most of you probably had a vaccination for the chickenpox, but if you had the chickenpox when you were a kid, you never usually get it again. And the reason is that you have that memory chip for chickenpox and your immune system knows what to do. And you may have been exposed, 60 times to chickenpox since you had the chickenpox, but you never get it. All right. The vaccine works the same way. You did never have to get it, but you now are immune to it because it knows you've primed your immune system. You've taught your immune system what to do. The picture on the right-hand side is showing you the difference between how quickly your immune system ramps up when you have primary and secondary exposures. So you have the primary immune response the first time around, and you see how much antibody you make. And then you have then you get well, and then you have a secondary exposure to it, and you see how quickly your body ramps up that secondary immune response so that you don't even know that you were exposed. So let's talk a little bit at our cells that we have in adaptive immunity. We have our B cells or our B lymphocytes. They're called B cells because they mature in the bone marrow, right? They make antibodies, and we're going to talk about that a lot more in upcoming up here in a few minutes. The old name for this is humoral immunity. The T cells or T lymphocytes mature in the thymus, so that's why they're called T cells for thymus. They are what orchestrate or coordinate the innate and adaptive responses to something that you have encountered. And the name for this one, the old name for this one, was cellular immunity. B cells attack invaders outside of the cells. T cells attack invaders once they have gotten into a cell. And so the picture that you see up there, the micrograph that you see, looks like to me that it's probably a lymphocyte of some sort. We can't tell which one it is without a different type of testing. So again, T cells are called T cells because they mature in the thymus. B cells are called B cells because they mature in the bone marrow, right? So they're both made in the bone marrow. The pre-T cell is going to leave and go to the thymus where it's going to finish its maturation process. The B cell is going to originate in the bone marrow and it stays there and matures there. So that's the difference between the two, is where do you mature? So we have lots of different kinds of lymphocytes. We're going to be talking about these: natural killer cells, CD8s, CD4s, regulatories, B cells. Antigens. The immune system evaluates cells by examining molecules on their cell surface that are called antigens. So an antigen stands for antibody generator. These are unique to specific pathogens, and they can be carbohydrates, lipids, nucleic acids, or proteins. Some examples include the cell walls of fimbria, the bacterial toxins, enzyme, virus spikes. I think about antigen like a flag. So if you've ever played flag football, or you know anybody that plays flag football, you basically have a flag you carry in your pocket, and that's how you tell who's on what team. That's how these antigens, that's what they remind me of. They remind me of a flag. They basically alert the immune system as to what they are. All right. Epitopes are an area of an antigen that the T cell can interact with. So they're showing you that picture. The whole thing is the antigen. The epitopes are the part that the T cell is able to connect to. So antigen antibody binding. So a typical protein antigen has multiple epitopes, and they're showing you that, all the different little spiky things on there. They're shown by the ability in this picture of 3 different antibodies to bind to the different epitopes of the same antigen. So the antibodies are what your B cells make. The antigen is what has infected your cell. And so the goal is for the antibody from your immune system to bind with the antigen on the bad guy, on the bacterial cell, so that it can destroy it. And when it binds to that, they call that antigen antibody binding. Haptens are small foreign molecules that are too small by themselves to cause an immune response. Some examples of haptens are drugs like penicillin, metals, ordinary, innocuous, household, industrial, environmental chemicals, things like that. If this incomplete molecule is linked to a larger carrier molecule, however, the combination develops immunogenicity. The hapten serves as the epitope. So I've said the word antibody a few times now. Let's talk about what it is. These are also called immunoglobulins, and that is abbreviated capital I little g, and then it'll have another capital letter after it because there are several different antibodies. What they are glycoproteins that are found in your blood and your tissue fluids, right? They come from our B cells, right? Specifically the plasma cells that the B cells make. They are composed of a heavy chain and a light chain. They have a Fab region, which is a fragment of an antigen binding. They have an FC region, which is a fragment of crystallization and the site of complement factor binding, and the site of binding to phagocytic cells. So you can see what one looks like over there on the right-hand side. Sorry if you hear my cat purring. It's Oscar. So, as I mentioned, there are several different classes of antibodies, and this is something that you want to make sure that you know, right? And so, remember I said it's capital I, little g, and then a capital letter, all right? So, you've got IgA, IgD, IgE, IgG, and IgM. What I want you to know is the properties, where you find these, what they do, that kind of thing. I could care less whether you know the structure of them or not, right? Nobody's ever going to ask you that. IgA is found in fluids. So mucus, saliva, tears, breast milk, it protects against pathogens. IgD is part of a B cell receptor. IgE activates mast cells and basophils, both of which release histamines, and they protect against parasitic worms. They're responsible for allergic reactions. IgG is secreted by plasma cells in the blood. This is the one antibody that is able to cross the placenta to the fetus. So mom can pass this over. So you might have heard that mom gives her immune system to the baby. This is what they're talking about. So IgG is the immunoglobulin or antibody that can pass over. It accounts for 80% of the circulating antibodies. And then IgM may be attached to the surface of B cells and secreted into the blood. It's responsible for the early stages of immunity. So those are the two things that I want you to know. The word gamed, if you use that word, it'll help you remember GAMED, which is all of the different immunoglobulins. So how do antibodies function? This is that same idea. At the end of the day, we looked at this in the last chapter. At the end of the day, the goal is to get rid of the antigen. And so it's going to do that one way or another. So it can do that through neutralization. This is when we bind the antibody, either IgG, IgM, or IgA, to the surface of the epitope or to the toxin, and it prevents the attachment of the pathogen to the host. All right, so that's one way that we can get rid of that antigen. Remember, the antigen is anything that your body deems as foreign. In this case, we're assuming that it is a pathogen, right? And so it's floating around in your blood, and one of those antibodies attaches to it, and that keeps it from going after the host cell. Opsonization is coating. Remember, this is the sugar coating that I was talking about before. IgG is a good opsonin, and it helps the phagocytes ingest the pathogens. So it basically puts this nice crunchy sugar coating on the outside. It's not really a sugar coating, but we're just going to pretend it is, and it makes it attractive to the phagocyte so that it goes after it. Agglutination. Or agglutination. This is when we have cross-linking of the pathogens. This is conducted by IgG and IgM. So you can see in the picture, we have our little red things, and we have our antibodies, which are the little spiky things. The red things are the bacteria. The spiky things are the gold things. Those are our antibodies. And so we basically cross-link those, and we form an aggregate, and phagocytic cells like those better. So they get a bigger meal, right? It's like getting a whole Happy Meal instead of just a hamburger. The aggregates are easier for the kidneys and the spleens to filter from the blood as well. And complement activation. Remember, complement is that series of proteins that's floating around in the inactive state that helps the immune system do its job better. IgM and IgG are responsible for this. This allows for the recruitment and activation of C1 or complement 1 complex. So that's another thing that antibodies can do. Antibody-dependent cell-mediated cytotoxicity, ADCC. This enhances the killing of pathogens that are too large to be phagocytosed. IgG does this. It brings the pathogen in close proximity to the natural killer cells. Natural killer cells are part of the innate immune system. And remember, they're also able to engage in that process of releasing perforin, and that punches holes in the cell membrane of the pathogen. So those are all the ways that antibodies do their job. So the end of the day, the job or the goal of the antibody is to eliminate the antigen. So one of those 5 ways is going to do that. All right. Let's talk a little bit about major histocompatibility complexes and antigen presenting cells. So again, our objectives. So all cells have a variety of flags or markers on their surface. Those are used for detection, they're used for recognition, and they're used for communication. Those markers play important roles in activating different components of immunity. CD molecules, CD stands for cluster of differentiation. These are cell surface markers. There are over 300 CD molecules that have been identified and named. Many of them are involved in the immune response. So what about lymphocytic receptors? The major role is to accept or grasp antigen in some form. Remember, antigen's the bad guy, right? So that is the goal, is to grab one of those antigens and get them out of circulation. B cells have receptors called BCRs that bind the antigens. T cells have receptors called TCR that bind antigens that have been processed and complexed with an MHC molecule on the presenting cell surface. Receptors have the capability or capacity to respond to nearly an infinite number of different antigens. So here's the idea. B cells, they find an antigen, they just go after them. T cells are only able to go after antigens that have already been made their way into a normal healthy cell. And so when that happens, the normal healthy cell puts up a flag on the surface and basically says, I'm infected. All right. That's what they mean complexed with an MHC molecule. So the MHC marker on that cell is going to bind with the marker off of that antigen, and basically it's a flag out on the surface to the immune system that says I'm infected. Because how would you know if the antigen is already inside of the cell? How would the immune cell know to go after that particular body cell? It wouldn't know because it's hiding the antigen inside of it. So it flies that flag out on the surface. All right, major histocompatibility complex molecules. There are two kinds. MHC1, which is found on all nucleated cells. This presents normal self-antigens and abnormal pathogens to effector T cells. And MHC2, this is found on macrophages, dendritic cells, and B cells. These present abnormal pathogen and abnormal pathogen antigens for initial activation of T cells. So antigen presentation. In most immune reaction, the antigen must be formally presented to lymphocytes by antigen-presenting cells called APCs. Some examples of APCs include macrophages, dendrites, and B cells. After processing is complete, the antigen is bound to the MHC receptor and moved to the surface of the APC so that it is readily accessible to the T lymphocyte. So that same idea that I was talking about before, when a cell processes or ingests that particular antigen, it's going to strip its MHC receptor off because it doesn't match the MHC receptors that are in the body, and move it out to the surface of its own cell, which basically tells the T cells that cell has been infected. All nucleated cells are capable of presenting antigens, but only macrophages, dendritic, and B cells can activate T cells. Antigen presentation with MHC2 molecules. The APC recognizes and attaches to the pathogen. The pathogen is engulfed by phagocytosis and then broken down. Antigen processing and presentation occurs, and only most antigenic epitopes are presented. So it's showing you in the picture, the bacteria is engulfed by phagocytosis into a dendritic cell and is encased in a phagosome. The lysosome fuses with that phagosome and digests the bacterium. That's the normal process of phagocytosis. And then the immunodominant epitopes are associated with MH2 and are presented on the cell surface. It basically puts a flag out on the surface and says, here's who I captured. Antigen presentation with MHC1 molecules. The healthy cells present normal self-antigen epitopes. These bind with the MHC1 binding cleft, and the immune cell, such as a natural killer cell, is going to recognize it and targets the cell for destruction. Infected cells, antigens specific to a particular pathogen are displayed on MHC1. This signals the natural killer cells or the another group of T cells to destroy the infected cell, and the T cell must first be activated before that can happen. So the moral of the story is there's lots of ways to activate the immune system. All of it involves the flag or the MHC marker off of the bacteria that's a bad guy, and that being presented on the cell surface of a healthy cell so that the immune system knows what's going on. A little about T cells and cellular immunity. Again, our objectives. All right, just kind of showing you all of the different kinds of T cells that we have. We got more kinds of T cells than we did B cells. B cells, we basically just had two kinds, the T cells and the B cells. But when we start talking about, I'm sorry, I meant humoral. We start talking about T cells, there's a lot of different flavors of T cells. So T cells, again, are produced in the red bone marrow. They come from a lymphoid stem cell. They differentiate into immature lymphocytes that we call thymocytes. They go into the bloodstream and they travel over to the thymus. That's where they're going to finish their process. Thymocytes are immature T cells, T lymphocytes that are in the thymus. They go through the process of thymic selection. It is a positive and negative selection process of the T cells. This is an essential mechanism that has to happen to generate the mature T cells, committing them to being a CD4 or CD8 lineage and avoiding autoimmunity. So basically, what happens is they test them. The thymus tests them to make sure that they're not going to activate on a cell, a cell that normally belongs in the body. 98% of the thymocytes get eliminated. Only 2% pass the test. If they are released and they are not selected correctly and they have the ability to activate the immune system on a harmless subject like one of your regular cells, that's the basis for autoimmune diseases. Autoimmune diseases are when your own immune system is attacking your own cells, and that's because T cells didn't go through this process of thymic selection correctly. So here's a little bit more on the classes of different T cells like I was talking about. You have your helper T cells. Their surface CD molecule is called a CD4. They are activated by presenting antigens associated with MHC2, and their function is to make everything happen. They're the helper. They orchestrate the two different sides of the immune system, the humoral and the cellular immunity, and they are involved in the activation of both macrophages and natural killer cells. Regulatory Ts, they used to be called suppressor Ts, so you might see that occasionally. These are also have a cell surface molecule called a CD4. The antigen presenting cells are going to be associated with MHC2s as well. These guys have a different function. They're involved in peripheral tolerance and prevention of autoimmune responses. And basically what they do is they reduce the number of B cells, specifically plasma cells, after the battle has been won. So when you have an infection, your immune system ramps up and makes a whole bunch of cells that will fight, right? And once that battle is over with, you got to get rid of all those extra warrior cells, because if you don't, they're going to attack something and they could attack your own tissues. So this is what the regulatory T cells do. And then the cytotoxic T cells, they have a different surface molecule, a CD8. These have a presenting cell associated with MHC1. These destroy cells that are infected with intracellular pathogens. So these go after things that have already gotten into a cell, like a virus. T cell receptors, TCRs, these are involved in pathogen epitope recognition. They have a variable and a constant region. They are epitope specific. There are 25 million T cells with unique epitope binding TCRs. This variability is actually due to your genetics. So you inherit a lot of your immune system from your parents. So this is what the surface of T cells look like in the imaginary world, right? They have all kinds of different flags and markers and all sorts of things going on out there. So you can see all of that, which would be on the typical T cell surface. So T cell reactions are the most complex and diverse in the immune system, and they involve several subsets of T cells. It involves the helper Ts, the cytotoxics, and the regulatory Ts. The actions of the T cells are dictated by the antigen-presenting cells that activate them. They are restricted. They require some type of MHC or self-recognition before they can be activated. All T cells produce cytokines with a spectrum of biological effects, but they do not produce antibodies. Antibodies are only produced by plasma cells, and plasma cells are produced by B cells. The end result of T cell stimulation is a mobilization of other T cells, B cells, and phagocytes. So T cells are the one that really sets off our immune system. So when there's a problem with the T cells, then there is a problem with the immune system. And so that's exactly what you see in situations like HIV/AIDS. In HIV/AIDS, the HIV virus selectively takes out the helper T cells. The helper T cells are the town crier, essentially. They're the one that alerts the rest of the immune system and goes into action. And so without them, you have no way of alerting the immune system, which is why people with advanced HIV/AIDS end up dying from diseases that and illnesses that your immune system typically could take care of, but for them, their immune system doesn't even know it's there. All right, the three types of helper T cells all bear the CD4 marker and are critical in regulating immune reactions to antigens. Helper T cells do not directly kill infected cells. Instead, as I mentioned, they activate the cytotoxic T cells and macrophages to attack the infected cells, or they stimulate the B cells to secrete antibodies. Their name tells you exactly what they do. They are the helper. Helper T cells become activated by interacting with antigen-presenting cells such as macrophages. The steps to this include TCR recognition of the foreign epitope, interaction of the CD4 and the MH2 with the MH2 molecule. Antigen-presenting cell and T cells are going to secrete the cytokines that are going to activate the helper T cell, and then proliferation occurs. Cytotoxic T cells are the cells that kill other cells. These are activated by antigen-presenting cells in 3 steps, similar to what we see with the helper T cells. The difference is this involves an MHC1 marker rather than an MHC2 marker. Once activated, they kill infected cells. They do this using perforin and granzyme. So that these are a group of cells like the natural killer cells that punch holes in the cell membrane of the cell they want to get rid of. The target cells that they target cells that the cytotoxic Ts can destroy. These include things like virally infected cells. They're able to recognize the virus peptides that are displayed out on the cell surface when the cell is telling you that it's infected. Cancer cells, these are constantly surveying the tissues and immediately attack any abnormal cell they encounter. The cells from other animals and humans, this is the most important factor in why we see graft rejection, so organ rejection, is because the cytotoxic C cells don't recognize the cells that have been brought into the body. The regulatory T cells, their name suggests what they do. They are regulatory. These cells have a role in regulating or suppressing the other cells in the immune system, which is their old name, again, the suppressor T cells. They control the immune response to self and foreign particles, which we call antigens, and they help to prevent autoimmune disease. Superantigens and unregulated activation of T cells. Unregulated and excessive activation of T cells can be life-threatening. Some examples of bacteria that cause this is streptococcal enterotoxins, streptococcal pyogenes toxins, and streptococcal mitogenic exotoxins. This can result in what they call a cytokine storm, which is an excessive, overblown inflammatory response. So that's how our T cells work. Complicated. It's really complicated. B cells and humoral immunity, let's talk about how they work. And of course, we have our objectives. So this is what we're going to be looking at here, how the B cells do their job. It's a little less complicated. So again, B cells are produced in the bone marrow, the red bone marrow specifically, but they don't leave and go anywhere. They just stay right there in the bone marrow and they mature there. In the maturation process, they are assessed for their antigen binding receptors. Any of the B cells that are self-reacting, just like the T cells that don't go after the right thing, they go after self cells, they're eliminated. Those that pass the selection travel to the spleen, and they do their final maturation in the spleen. And at that point, they are released and become naive, mature B cells. B cell receptors, or BCRs, IgD and IgM, are what we're talking about here. They have identical heavy and light chains. There are up to 100,000 BCRs on each naive mature B cell. These are activated, so the B cell functions as an antigen presenting cell. The proteins are T-dependent antigens. The non-protein antigens are T-independent antigens. So here's what the imaginary surface of a B cell looks like. Not so very different than what we saw with the T cell, really. So how do B cells respond to antigen when they are activated? B cells don't come in contact with the antigen directly like we saw with T cells. T cells will bind it. T cells will do all kinds of things. B cells, on the other hand, don't want to get their hands dirty, so they start to multiply rapidly and produce what are called plasma cells. And it's the plasma cells that release the antibodies. Remember those 5 gamed antibodies that we talked about into the tissue in the blood, and it's the antibodies that attach to the antigen for which they are specific, and the antigen is marked for destruction or neutralization. So a little bit different process, takes a little longer. All right, B cells are capable of being activated without a T cell, okay? So B cells that do this are activated by the polysaccharide capsules, the lipopolysaccharides, and cross-linking of multiple BCRs. A second activation signal has to occur. These are toll-like receptors and PAMPs, and the complement system can do this as well. Once activated, they proliferate differentiated plasma cells, which are antibody-producing cells. So the idea is this: we don't want those B cells to just randomly fire off and start making plasma cells. So there has to be 2 activation signals. 2 activation signals. When B cells are activated with T cells, it is a stronger immune response, and we get those lovely memory cells. Free protein antigens can do this, and protein antigens associated with the pathogen can do this. The BCR interaction with the free protein is going to stimulate internalization of the antigen. It's processed and presented with the MHC2 marker, and it's recognized by the helper T cells, and the helper T cells get things going. Linked recognition, this is when the T cells' CD4 molecules interact with the MHC2 on the B cells. The activated T cells, the helper Ts produce the cytokines. These activate the B cells and cause proliferation into memory B cells and plasma cells. IgM is secreted. There is class switching that can happen. This is when the helper T cells are going to stimulate the plasma cells to switch from IgM to IgG, IgA, or IgE. This allows for a variety of different antibody classes to be produced. The variable region remains the same, so they have the same specificity. So that's an important idea that, different plasma cells, you would think there would be one plasma cell would only release one kind of antibody, but that's not true. They're able to switch and start releasing something different, because remember when we looked at that list a while ago, that the antibodies, they do different things. Your primary response has a lag period of about 10 days. So you've been exposed to something. It takes about 10 days. There's no antibody is detected in the serum. This is the time that's required for all the steps of the primary response. There's a rise in the IgM signal that ends at the lag period as the B cells differentiate into antibody producing cells or plasma cells. The IgM levels go down and the B cells produce IgG and through class switching. The secondary response that we talk about occurs much quickly and forcefully. The lag period is only a few days, and IgG production is much higher. So there's that idea. The first time you come in contact with a novel antigen, you unfortunately are going to get sick, and there's going to be a lag period, and it's going to be a long period of time before you get well. But the second time around, your immune system already knows what to do because it has memory cells, and you're able to fight that battle without even knowing it. So you might have had this response before. You get up one morning, and for whatever reason, your lymph nodes are swollen in your neck. and you think, oh no, I'm getting sick. But then it goes away. That is a secondary response. So when your lymph nodes swell up, the lymph nodes are loaded with all kinds of T cells and B cells. And when you come in contact with something out there, they multiply like crazy. And what you feel is the swelling because they're getting ready to start filtering that lymph as it travels back to the blood of all of the antigen it can find. Well, when you come in contact with something you already have a memory chip for, it automatically makes those lymph nodes swell up, fights the battle, and you get well. So you might have swollen lymph nodes for a couple of days, but you never get any sicker from that. That's just evidence to you that your immune system is working. So again, just showing you the difference between a primary and secondary immune response. So here's a nice summary chart or review table of the contrasting properties of B cells and T cells. So what's the difference between T cells and B cells? So this is a really, really great table to review. So let's talk a little bit about the highly controversial right now, vaccines. Again, some objectives. Natural immunity, all immunity that is acquired through the normal biological experiences of an individual. Artificial immunity is protection from an infection obtained through medical procedures like vaccines or immune serums. Active immunity occurs when an individual receives immune stimulus that activates the B cells and the T cells to produce immune substances such as antibodies. Active immunity is what creates memory cells that render the person ready for quick action anytime you're re-exposed to that same antigen. It requires several days to develop. It lasts for a relatively long period, sometimes a lifetime, and it can be stimulated by both natural and artificial means. Passive immunity occurs when an individual receives antibodies from another human or an animal. Passive immunity is characterized by the recipient being protected for a short period of time, even though they have not had any prior exposure to the antigen. The lack of memory for the original antigen and the lack of antibody production against the disease. This has immediate onset of protection. This is short-term, has short-term effectiveness, and it can be natural or artificial. So passive immunity is what mom is passing over to the baby through the placenta, and it's also what she's passing over to the baby with breast milk. All right. This is also if you have to get an immunoglobulin injection because you were exposed to some particular disease. A good example of this is hepatitis A. So if you've ever been exposed to hepatitis A, they tell you you have to get an immunoglobulin or a gamma globulin shot. And the reason what they're giving you is they're giving you the immunoglobulins. They're giving you the antibody a little bit of time, about 6 weeks, to make the proper antibodies to fight hepatitis A. And in that 6 weeks, hepatitis A can do a lot of damage. So they give you the antibodies so that you have a bridge until your own immune system has time to make them. All right. That's considered passive because you're not making them right away. We're giving you what you need so that you can fight the disease or the illness without getting too sick. All right. So different ways that we talk about the immune or our immunity. Naturally acquired and artificially acquired, right? So naturally acquired means you've been exposed to it. Artificially means that you've been you gained it through another individual or an animal, right? Passive means that you're passing the antibodies like breast milk through or through the placenta, right? So passive means we're given the antibodies, or if we get a immunoglobulin shot, right? Active is when we actually make the antibodies. So the immunity through illness and recovery, you get the chicken pox, right? And so you are now immune to the chicken pox, or you get a vaccine. What we're trying to create is herd immunity. This is a reduction in the prevalence of a disease brought about when a few individuals in the population are susceptible to an infectious agent. Vaccination programs greatly reduce the number of susceptible people in a population. The herd is protected because there are too few susceptible individuals in the population for the disease to spread effectively. Those that are not or cannot be vaccinated because they have some reason, medical reason, immunocompromised or whatever, will be unlikely to encounter the pathogen. You basically want to keep it from spreading, and you keep it from spreading by immunizing as many people as you possibly can. So looking at it with cats, you can read through all of that. You can see why we want a population to get immunized. If we get our population immunized, we cut down on the spread of disease. There's always going to be a few individuals that are not immunized for whatever reason, right? But hopefully enough people are that we are protecting everyone else. Viriolization or viriolation. This is inoculation with infectious material from scabs or pustules. The infectious materials are either injected into the skin or introduced through the nasal route. This can be pretty risky. Sometimes serious or fatal infections can occur. They did this originally with smallpox. They don't do this with smallpox anymore. They don't even give you smallpox immunizations anymore. But this was how we originally were doing giving people immunity against smallpox. Vaccination is how we do things now. This is inoculation with a less virulent pathogen. So you're either given a killed or attenuated version, and that means that it has been weakened. Attenuated means weakened. It can't make you sick. This is attributed to Edward Jenner. He was the one that developed the process of vaccination. These was inspired by dairy maids who had been infected by cowpox when they were milking. Cows get a particular pox called cowpox, and what they noticed was that people who milked cows for a living and ended up with a mild case of cowpox, because it didn't make you very sick, never got smallpox. So he tested his theory by injecting a young boy with material from the human cowpox lesions and exposed him to smallpox 2 months later, and the boy was immune. So he actually, the young boy he's talking about was actually his own son. The use of vaccines was first regarded with fear and skepticism, but later adopted when it proved to be successful. Vaccines are highly successful. We know that. We have tons and tons of data that says that vaccines are safe and that vaccines are the way to go. We do not want to not vaccinate animals. Can you imagine what would happen if we stopped vaccinating for rabies? We would have all kinds of people who ended up with rabies because they got bit by rabid animals. We absolutely positively don't want to stop vaccinations. So here are some different classes of vaccinations, and this is something you want to make sure that you know. You want to read through this. So we have the different classes in the first column. We have a description of what that means, and then we talk about what's the advantage and what's the disadvantage, and then some examples. So you definitely want to know what a live attenuated means, what inactivated means, what subunit means, toxoid means, and conjugate. And so make sure you know that. Make sure you know the advantages of the disadvantages and examples of them. All right, that is the end. I know that this immune stuff is really, really complicated. So focus on terminology, focus on steps, focus on things like different types of T cells, focus on things like uh, what's the end result? When we have that antigen antibody complex, what are we ultimately trying to do? Don't worry too much about the intimate details about how these things happen. It is extremely complicated and way beyond the scope of this course, quite frankly. It's graduate level type information. If you've got the big idea about the two sides of the immune system and examples of all of that and how they work and all that sort of thing, then you should be in good shape. And I will see you for the next lecture.